Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are studying now a chapter that is Principles of Inheritance and Variation, a class 12th chapter. In a continuation of this, we have continued and covered so many topics. And today, in this lecture, we will be continuing with the topic that is mutation. Okay. See, by hearing this term, what we can understand, so it is just explaining the something change. Okay, it is explaining about something change, but change in what? If that change occur in a genotype and phenotype, then we call it as a mutation. What we call mutation, when it is a change in a genotype or phenotype. And to show the change in a phenotype and genotype, there may be a rearrangement or change in a genes present on the DNA or the chromosomes, okay, or the number of chromosomes. In that certain phenomena, you will see the changes also. So, if we define this mutation, okay, this mutation, it is sudden, it is sudden, discontinuous, it is sudden, discontinuous variation in genotype and phenotype. When any sudden change or variation is observed in a genotype and phenotype of an organism, then organism due to change in chromosome and gene, chromosomes and genes, then it is known as mutation. So it is known that in addition to recombination, mutation is the another phenomena that lead to the variation in DNA. Means it is also one of the important role. Play also one of the important role in a what a particularly we can say a causing a variation. So in this mutation, we are going to study the different types. And first, we are going to study the type of mutation based on or depending on the cause. On cause of mutation, this mutation are of three types. And when this mutation are of three types, they are defined as first, that is gene mutation. Another one is the chromosomal aberration. And the third one is the genomic mutation. So these three type of mutation are seen: gene mutation, chromosomal aberration, and the genomic mutation. Okay, and these are the based on the cause of the mutation. So let's study all this mutation one by one. So when we first talk about the gene mutation, by hearing this term, we can predict that it is a mutation in the gene which are present on the DNA. Okay. So if we define a gene mutation, we can define it as It is alteration of DNA. It is alteration of DNA due to change in nucleotide sequence. So here the nucleotide sequence in gene is going to be affected. So here what happens, the gene which is present in DNA that get altered due to the nucleotide change in the nucleotide sequence. Now this change, if we say, or this mutation, that gene mutation may occur due to change in may occur due to change in single base pair, single base pair of DNA. So gene mutation may occur due to the change in single base pair of DNA. Then it is known as point mutation. And the classical example of this point mutation, if we talk about the example of this point mutation, then that is sickle cell anemia. And this sickle cell anemia we are going to discuss in the later part during the genetic disorders. Okay. But if change occur in more than one nucleotide pair, if change occur in more than one nucleotide pair, then it is called gross mutation. Then it is called a gross mutation. So two type of mutation are possible. One is point mutation is if it is affecting a single base pair, and it can be a gross mutation if it is affecting a particular multiple base pair or a nucleotide pairs. Now further, this gene mutation are divided into two types. They are of two types. Number one, frameshift mutation. Dear student, if we talk about frameshift mutation, then in this, if one nucleotide getting added or one nucleotide getting removed, it is changing a frame of reading, a reading frame of the codon. And if reading frame of codon is getting affected, then the sequence of amino acid in the protein is going to be affected. So we call it as a frame shift mutation. What do we call? A frame shift mutation. So in this frame shift mutation is again of a two type. One is deletion, and deletion is defined as the removal of one or more bases from nucleotide chain. If there is a removal of one or more bases from a nucleotide chain, then we call it as a deletion. See, suppose there is a nucleotide sequence on the DNA. We are taking an example. Is A T G C C G T A C G C A. Okay. Now this is a 3 dash 2 5 dash and on DNA. Now when it getting transcribed as a mRNA, on mRNA it is going to UAC, GGC, AUC, CGU. Such sequence is going to be expressed on the mRNA in 5 dash to 3 dash direction. Now, when we talk about the protein or polypeptide which is coded or translated by this mRNA, then this three nucleotide code for a one amino acid, another three nucleotide code for another amino acid, another three nucleotide code for another amino acid. Okay. And if they are coding for the different amino acid, it is known that it is known that the particularly UAC, UAC is the first codon which is coding for the tyrosine amino acid. Okay. Then GGC. Okay. So GGC is coding for the glycine amino acid. AUC. Then AUC code for the isoleucine amino acid. And CGU code for a arginine amino acid. See. Now if the deletion occur of this base get deleted, deletion of Z. So now the sequence become like this. If the same G get deleted here or C get deleted, actually C get deleted, then G will be deleted here. Okay. So if C will be deleted on the DNA, now the coding sequence is going to be like this: ATG, then CGT, ACG, CA, and 5 dash. Same way. So mRNA is going to be 5 dash UAC, GCA, A complementary is U, C, UGC, and C complementary to G. Okay, so it is GCA, UCC. Okay, so here this GCG. Okay, so ATG, the USC, CG, GC, AU, CG, GC, CGAU, and this 3 dash end will occur. Now, when it called for polypeptide, this UAC coding for the tyrosine, but later on GCA, if we talk, then GCA code for alanine. Then now the coding frame is listen. Then UGC, UGC, if we talk, then it is going to be cysteine. And then GU and the particularly anything can be there. Then GU, and if any UCAG, it will be coding for the valine. So you can see by the just a deletion of this C, the whole three amino acid in a polypeptide getting changed. Why? Because the coding frame of a triplet codon is changed. And when a coding frame of triplet codon gets changed, due to the deletion of a single base pair, the whole particularly what a polypeptide sequence of amino acid is getting changed. So in this way, it is known as frameshift mutation. Now, after frameshift mutation, in frameshift mutation, one is we have discussed that is deletion. Same way, another one is the substitution. Another one is the insertion or addition. So B is insertion or addition. If we talk about insertion or addition, then in this there is a addition of one or more bases in a nucleotide chain. Okay. Now, if here addition of one or more bases occur in a nucleotide chain, then it is known as insertion or addition. So same way, if there is a DNA and the DNA sequence is ATCGCGATACG like this, G A, and it is a 5 addition. So the mRNA is going to be like this. Okay, let's change the sequence and draw it on a separate side so we'll have space to make it properly. I'm taking here the DNA sequence. It is 3 dash ATGCGCATCGAC. Anything you can take. Okay, okay, and it is a 5 dash. Now, if we code for its mRNA by transcription, then mRNA is going to code for UAC, then GCG, UAG, CUG, and it is 3 dash. It is mRNA. Now, if we code for a polypeptide protein or polypeptide or amino acid sequence, then in amino acid sequence, you will see that UAC that is going to code for the tyrosine, then another codon that is on the mRNA that is GCG, then GCG code for alanine, then UAG codon for code for the what? A stop codon. So here, so let's change this somewhat nucleotide sequence. So it will be not a stop codon because UAG is going to be stop codon in mRNA. So further polypeptide will not synthesize. So here I am replacing this ATC with the G. Okay. So I am writing ATG. So here it can be UAC. And UAC code for again tyrosine. And CUG. If you talk about CUG, it is going to be code for leucine. Okay. This four amino acid getting. Now if here in DNA, this particular the G on G you will see here T is getting inserted. So now the sequence is going to be ATT, then GCG, CAT, GGAC, and 5 dash. Now, if we talk about mRNA, in mRNA you will see 5 dash ATT. So UAA, UAA, UAG, UGA. So it is going to be stop codon. So we can't write UTT. Here I am changing. I am inserting a U. I am inserting here A. Okay, so U A. Okay, see, I am removing this and I am not inserting here, but I am inserting here C. I am inserting here C. Okay, so here it is going to be in place of ATT, it is going to be ATC. Now ATC, then A code for U, A complementary to U, T complementary to A, C complementary to G, UAG. Again, it's going to be stop code. So to what I should do, what I should do, A. I can insert A. Okay, I'm inserting A. So if I insert A, see, let's see what happens. If it is I insert A and here A, then it ATA become UA U. Okay. Then G will be complementary to C, GC, GU, A, CC, U, and G. This will be codon. Now this is going to be the first codon. This is second. This is third. This is fourth. Now due to the what insertion, the UAU is going to code for the tyrosine. Okay. Then CGC, CGC is going to code for the arginine. GUA, GUA is going to code for valine. And here this, sorry, this is going to be codon, that is a CCU. So CCU is going to be proline. Okay. So here, if you see, due to the insertion of just one A at this position, the reading frame of further three amino acid is getting changed. So due to insertion or addition of a nucleotide in the nucleotide chain, the particular whole reading frame of mRNA is getting changed due to the change in DNA, and same way due to this the amino acid in polypeptide are also getting changed. So this way, insertion or a deletion will affect the whole particular polypeptide chain and it can cause a mutation. So it is a frameshift mutation. After frameshift, we are going to talk about another type, that is a substitution. That is a type of gene mutation. When we talk about substitution, in substitution mutation, what happens? The replacement of one base by another. So here we are going to talk about replacement by one by the another. Now it is of two types. One is transition and another type is transversion. Now in transition, the purine base is removed and in place of that only purine is getting incorporated. Or if the pyrimidine base is removed and it is replaced by pyrimidine base only, then we call it as a what? Transition. So in transition, what happens? The purine base, that is adenine or guanine, is replaced or substituted by another purine basis or pyrimidine base, that is T or C, is substituted by another pyrimidine base, then it is known as transition. So in transition, what is happening? If a DNA is there, in DNA purine is there, either it is A or G. If it is A, then it is replaced by G, and if it is G and that is replaced by A, then we call it as a transition. Or in a pyrimidine case, if it is a T in the DNA mutation, it is converted into the C. Or if C is there in DNA, it is converted into T. If such a type of mutation is occurring, then we call it as a transition. If I want to write this in a short way, I can write like this, that adenine and guanine are replaced like this, and here thymine and cytosine are replaced like this. But in transversion, if you see in this, the substitution of purine base with pyrimidine base or vice versa, such substitution if occur, then it is known as transversion. And in this, if suppose purine is there and purine is adenine, then adenine can be changed by the thymine, or if adenine is getting changed by the cytosine, or cytosine is changed by the A and thymine changed by the A, then it is known as what, a particularly a purine to pyrimidine, pyrimidine to purine conversion. Or we can say, if guanine is changed to the thymine, or thymine is changed to the guanine, or guanine is changed to the cytosine, or cytosine is replaced by the guanine, then it is known as what, substitution. So this four combination can occur in case of transversion. Either A is replaced by T, A is replaced by C, T is replaced by A, C is replaced by A, G is replaced by T, or T is replaced by G, G is replaced by C, or C is replaced by G. Then in a DNA or a nucleotide sequence, then it is known as what, a particular transition and transversion. Okay. So this is the thing which occur. Now this gene mutation, it is known that they may occur naturally or automatically. We remember, gene mutation may occur naturally or automatically due to internal regions. So either it is naturally or it is artificially by the human interference or by the particular any agent interference. So if occur naturally or automatically, here it is due to internal region, then they are named as spontaneous mutation. They are named as spontaneous mutation. And if if it is produced by, or gene mutation is produced by external physical or chemical factor, then these are named as mutagens, and the mutation is called as induced mutation. So if it is occurring by the internal region, any natural automatic region, then it is, if it is a DNA replication is going on, and an inappropriate complementary base pair is incorporated, then it is spontaneous mutation. But if any chemical goes and carry out such change, then it is known as what? Induced mutation. Why we are using it? Inducing. It is not occurring naturally. We are inducing it. So it is known as induced mutation. Okay. So this is all about the gene mutation. Now after gene mutation, we are going to talk about the another type of mutations are known as chromosomal aberration. So let's talk about chromosomal aberration. It is chromosomal aberration. When we call it as a chromosomal aberrations, dear students, we know that chromosomes are usually made up of protein, DNA, and RNA. And in chromosome, there are chromatids, two chromatids combined to form a chromosome. And in each chromatid, you know that they have a one DNA helix that run continuously from one end to another end. In chromatids, DNA present in a highly supercoiled manner. See, if this is the chromosome, this is a chromosome, and in this DNA, you know that it is highly coiled in nature. And this has a this one chromatid. This whole is considered as one chromatid, and this whole is considered as chromatid. So if I draw like this is one chromatid, and the remaining one is the another chromatid. Now in this one chromatid, if you see, the DNA helix run from this end to this end, and then the DNA in this chromatid is in a highly supercoiled form. So it has DNA in highly supercoiled form. So when it is present in highly supercoiled form, the loss or gain of a segment of DNA result in alteration of chromosome. Okay. So loss or gain. Loss means deletion, or gain means addition or insertion or duplication of a segment of DNA cause alteration in chromosomes. And this alteration in chromosome, if we talk, We also know that genes are located on chromosome, means by DNA, and DNA is in a chromosome. So that alteration in chromosome result in chromosome abnormalities or the aberration. Why? Because the whole structure of chromosome is going to be affected. So obviously genes are going to be affected. Okay, and thus this type of chromosomal aberration are commonly seen in case of the cancer cells. Now the question is, which type of this chromosomal aberration are seen? So there are four types of chromosomal aberration which we are going to discuss. So here the deletion or addition or a loss of a segment of DNA causes alteration in chromosome. So there might be a possibility of alteration of the genes which are present in this chromosome. Now it is of different type. So let's discuss the different type. The first type is deletion in case of chromosome. When we talk about deletion in case of chromosome, then dear students, it occurs when a part of chromosome is lost. If any segment of chromosome is lost, then it is known as deletion. Okay. So here a part of chromosome is lost. And when a part of chromosome is lost, this deletion of a chromosome segment can occur from the tip or can occur from the intercalary. So this deletion is of two types. Either this is a terminal deletion or intercalary or interstitial. Intercalary or interstitial deletion. Suppose this is the chromosome with a single chromatid. And on this, if the genes are present like this, A, B, C, D, E, F, and this terminal part is removed, suppose this A and B genes are removed, then the chromosome will look like this. This chromosome will have a C, D, E, and F. Means this segment get deleted. So the due to the loss of terminal segment of chromosome, the single break is produced here, and the particularly a chromosome get shortened. Okay, so the size of a chromosome get reduced due to this deletion. Now in intercalary or interstitial deletion, if we talk, this deletion occur in the middle position. So for that we can take example. Suppose this is the thing. This is A, B, C, D, E, F, and G. Okay. Now here, suppose this D and E get deleted. So the chromosome now will look like this. A, B, C segment remains same, but D and E get deleted, so F and G will become here. So here, in a terminal deletion, only one cut will be provided, So single break produced in the chromosome whereas in the intercalary or interstitial double break is produced in a chromosome. Okay, so this is the two type of deletion, that is terminal and intercalary. Now after this, the next type which is seen in case of the, we can say, chromosomal aberration, that is the second type, that is duplication. In duplication, if we see, here it occurs due to addition of a part of chromosome, so that a gene or set of set of gene is represented twice. So here, suppose this is the chromosome. In this chromosome, I have some genes like P, Q, R, S, T, U, V. Now in this RS get duplicated. So now the chromosome will look like this, which having the P, Q, R, S, R, S. Then T, U, V will be remain as S. So here you can see this RS and this RS getting duplicated twice. So it is known as duplication. Okay. Now after this, the third type, which is here, that is known as translocation. Dear student, in translocation, translocate means go from one place to another place. Yes or no? So this translocation obviously involves such shifting. So it is known that it involves shifting of a part of chromosome to another non-homologous chromosome. To another non-homologous chromosome, then it is known as a translocation. So new recombinant chromosome are formed, and this induce faulty pairing of chromosome. Faulty pairing of chromosome during meiosis. Okay, so if this is the one chromosome, this is having A, B, C, D, E, F gene, and this is the another chromosome which having a U, V, W, X, Y gene. Suppose this AB get deleted from here and transferred to here, then this chromosome will somewhat increase in size and will look like this: A, B, then U, V, then W, X, and Y. Whereas in this, if it is getting deleted, then this chromosome will be like this. If there is a presence of C, then D, E, and F. So this chromosome getting shortened due to the deletion, but this particular part is translocated here, so the size of this chromosome is going to be increased. So it is a translocation. Okay. Now, but one more type of translocation is there, which is known as the reciprocal translocation. One of the important ones. Reciprocal translocation or segmental interchange. In reciprocal translocation, actually what happens, dear students? Then here, mutual exchange of chromosome segment between non-homologous chromosomes. What happens here? Mutual exchange of chromosome segment between non-homologous chromosome. And when there is a segment exchange or mutual exchange, both chromosomes giving it to each other, then this is also known as illegitimate crossover. Illegitimate crossover. Okay. And this reciprocal translocation has evolutionary significance. So the question is how this reciprocal translocation is actually taking place. We need to visualize it. So let's visualize it in the next slide. Suppose this is the chromosome 22. Sorry, we can say any chromosome we can take. And this is another chromosome. Here they have A, B, C, D, E, F, G genes. Here they have U, V, W, X, Y, and Z. Okay. In this, suppose this A and V is getting deleted and it is transferred on this chromosome. And here, this U and V are getting deleted and they are transferred here. So this is the mutual exchange. So in this chromosome, you will see that A and B is getting deleted, but U and V are getting added. So now the chromosome will have U, V, C, D, E, F, G. Means this is the what a particular new segment which is added here. But in this, if you see this chromosome, in this chromosome also U and V is getting deleted, but A and B is added. The W, X, Y, Z remains same. So this A and B are a new segment. So up to this is the what a particular segment from the another chromosome. Okay. Here also this UV segment is from the what another chromosome. So this is showing a what a mutual exchange between these two chromosomes, the first and second chromosome. So it is known as what a particularly mutual exchange, or we can say a chromosomal translocation. Now, if we note down one example of translocation, then which causes cancer, then you should remember that one disorder is that chronic myelogenous leukemia. Myelogenous leukemia, that is CML. This CML occur due to translocation of segment of long arm from chromosome 22 to chromosome 9. So means the deletion is occurring on the chromosome 22 and the addition is occurring on the chromosome 9. So chromosome 22 is getting shortened, and this chromosome 22 discovered in which a Philadelphia city. So dear student, here you should remember that this chromosome 22 is known as Philadelphia chromosome. Philadelphia chromosome. Okay, you should remember this point with respect to this chronic myelogenous leukemia. Okay, dear students, so this is what about the translocation. Now we are going to talk about another fourth type of chromosomal aberration, and that fourth type of chromosomal aberration is inversion. If you talk about this inversion, what happens here? Here, change in linear order of genes, change in linear order of genes, change in linear order of genes by rotating a section of chromosome at 180 degrees Celsius, or at 180 degree, not Celsius, 180 degree. So if any segment in the chromosome is rotated at 180 degree, the completely inversion of this will occur. So here, suppose this is the chromosome and having a A, B, C, D, E, F genes. Now this B, C, and D, they are getting inverted. So now the chromosome will like this. That is A, then D, C, B, E, and F. So this D, C, and B are getting inverted. But you can see one of the important things there. Here a centromere is getting involved. If this occur in only A and B converted into the B and A, then there is no involvement of centromere. So it is of two types, that is inversion. First one is that paracentric. Paracentric means here inversion without involving centromere. So inversion is happening, but centromere is not involved. Then it is known as what? Particularly paracentric, or we can say inverted segment is not carrying the centromere. And second is pericentric. Pericentric means inversion involves centromere. Okay, the centromere, if it is involved, then it is a pericentric. Not involved, then it is paracentric. So such is known as inversion. And the inversion is known to frequently occur in case of Drosophila due to the X-ray. Okay, so this is the four different types which we have studied in case of the chromosomal aberration or a chromosomal mutation, and in that we can say the chromosome structure or chromosome structure is getting affected. A size may get affected, okay, due to this deletion, duplication, translocation, or inversion. Okay, now after this chromosome structure change, now we are going to discuss a third type of mutation in which you will see the change in chromosome number. Okay, and that we call a genometric mutation. So let's talk about the genometric mutation. So let's talk about the genometric mutation. Okay, so as I told you, this genometric mutation are going to deal with change in number of chromosomes. Okay, so here it is a change in chromosome number that about that bring about change in phenotype or any different phenotype. Actually, it is of two types. Number one is aneuploidy. If you talk about aneuploidy, then in this you will see that the chromosome number are somewhat 1, 2, or 3 plus to the total number of chromosomes, or they can be minus to the total number of chromosomes. So if any change in chromosome number change in chromosome number in an organism would be different than the multiple of then the multiple of basic set of chromosome, then it is known as aneuploidy. Means if we talk about the humans, in humans there are 46 chromosomes. If it becomes 47, 48, 49, or it becomes 44, then 44, 44, 45, 44, 43, like this, if the variation in the chromosome number is seen, then it is nothing but aneuploidy. Okay. Now this aneuploidy, it commonly arise due to non-junction of two homologous chromosomes. During cell division. So how this non-disjunction means the absence of a separation of two homologous chromosomes, means two homologous chromosomes are not going to separate in any one pair. So if you want to discuss this, let's discuss this. Suppose this is a cell which having a 3 pair, and this 2n is equal to 6. This one line shows the what chromosome. So total 6 chromosomes are there, and it is a diploid. Now, if non-disjunction take place, means in any one pair, if you don't find any separation in the homologous chromosome, because in gamete, this here two set are there. This one side of one each chromosome of this side and each chromosome of this side. So this will represent a separate set. So here two set are there. Now one set is known to be go here. Suppose these two are going, but the another chromosome of first pair is showing a non-disjunction. So it is going as a pair itself in this particular gamete. So here the total number of here it is diploid, so it should be haploid, and this should be also haploid. But here one chromosome is minus, so we write it as n minus one, and here one chromosome is getting increased, so it is known as n plus one. Why it is happening? Because these two homologous chromosomes are not getting separated easily during the particularly we can say division. So n minus one gamete and n plus one gametes are produced. And these are the two different gametes are produced when there is a non-disjunction take place. Okay. Now fusion of this gametes with the normal gamete give rise to the different type of aneuploids. So what happens now? The fusion of these gametes, that is n minus 1 or n plus 1 with normal gamete give rise to different type of aneuploids. Give rise to different types of aneuploids. Okay. So if here such fusion occur, then what will be the condition? Let's see. So if the haploid gamete, that is n, fuses with the n minus 1. So here this 2n will be there. This n and this n will be 2n, but one chromosome is less in this set. So it is going to be multiply. If you multiply, then it is going to be 2n minus 1. And if 2n minus 1 condition is seen, means suppose if 6 chromosomes are there in a diploid set, and here it is going to be 5. Okay. If it is, so we are talking about a human cell. In human cell, out of 46, one will be less. So it's going to be 45 chromosomes in case of human when this condition is seen. And this condition is known as monosomic condition. Same way, if this n minus 1 itself multiply with the another n minus 1, then it is going to be 2n minus 2. And when 2n minus 2 condition is there, then it is known as nullisomic condition. So according to nullisomic condition, if we talk about 2n is equal to 6 cells, then here 2 particularly whole pair is going, 2 particularly we can say whole pair is absent. So it is going to be 4 chromosomes. If it is a human cell and it has a 46 chromosome, then the nullisomic of it is formed, then it is having a 44 chromosome. Same way, if n multiply by with n plus 1, then it is going to be 2n plus 1. And one chromosome is going to be increased. So in place of 6, the 7 chromosomes are going to be take less. And in place of 44, 46 in case of human, 47 chromosome will be there. Then this condition is known as the trisomic condition. And if n plus 1 multiply with the n plus 1, then it is going to be 2n plus 2. Then it is what? A particularly trisomic condition. So if with respect to 2n plus 2n condition like this, 2n is equal to 6 and we form all variant of this, then 2n minus 1, that is the monosomic, you will see that there is 1 is absent. So here total number of chromosomes at 2n minus 1. So it is total number of chromosomes going to be 5. Okay, that's what that is. 2n minus 1 is monosomic or monosomic. So same way, if there is a whole pair is absent, then it is known as 2n minus 1. So it is going to be nullisomic. If apart from 6, any one chromosome is getting added to this one, that is known trisomic, that is 2n plus 1. And if whole pair getting added, then it is 2n plus 2, that is tetrasomic. Now in this case, it is also possible that these 2 chromosomes are not getting added to the one pair itself. One chromosome is getting added to the another cell. Then it is also going to be what? If total number of chromosomes are here 8, then in that condition also the chromosome number are going to be 8 like this. If this is a diploid, but one chromosome is getting added and one chromosome is getting added, then you will write here 2n plus 1 plus 1, and it will be referred as double monosomy. Okay, 2n double trisomy. Sorry. Same way, if 2 chromosomes are getting minus, here whole pair is going. But in that case, if one chromosome is going from the, suppose this is the cell, and in that one chromosome is going from the this 3 pair, one chromosome is going from first pair, one chromosome is going from the another pair, like this, then it is going to be 2n minus 1 minus 1. Then it is double monosomy. Okay, such condition can be observed during the aneuploidy condition. Now, if we note down some of the examples or disorders, we will discuss in the later part. But if we talk about some of the examples of aneuploidy, then in that, when trisomy occurs, that is 2n plus 1, some disorders are known to occur, that is Down syndrome, where a 21st trisomy occur, and the Klein filter syndrome. Then the second one is the monosomy. In monosomy, here see Y chromosome X chromosome getting placed. In male, only X chromosome character increase, so that is Klinefelter. But in monosomy Turner syndrome, one X chromosome getting reduced, so it will having the some of the male characteristics. Okay, so these are the some of the example of this. Now the another type of this genomic mutation is euploidy. And if you define euploidy, it is any change in number of chromosome. It is any change in number of chromosome in the multiple of the number of chromosome, or it is any change in the number of chromosome in the multiple of basic set of chromosome, or due to variation, or due to variation or due to variation in one or more haploid, in one or more haploid sets of chromosome. Now, this euploidy is again of two types. One is haploidy. In haploidy, one set of chromosome is there. It is haploidy means haploid organism. Presence of one set of chromosome. And it is known that these haploids are better for mutational experiment studies. Haploids are better for mutation experimental studies, because all mutation which occur in the haploid, either it is a dominant or a recessive, it is going to express immediately. All type of mutation is going to express immediately, because there is no case of dominance or recessiveness, because the another allele of any gene is not going to present on the homologous chromosome, because in haploids only one chromosome in present in out of each pair. Okay, so they are not chromosome are not going to be present in a pair. So alleles genes are not also present in the alleles. Okay, so here one allele of each gene present in each cell. So due to this, its expression or is occur immediately. So these are the best due to this. They are the best for the what mutational experimental studies. And another type. So we can write all mutation either dominant or recessive express immediately due to the what? Presence of a single allele of each gene in each cell. And the second type of this euploidy is polyploidy. And when we talk about this polyploidy, it is the presence of more than two set of chromosomes. It is defined as presence of more than two set of chromosome in a cell. Then it is known as polyploidy. Now, why it happens? Because failure of cytokinesis after telophase. After telophase stage of mitosis, a failure of cytokinesis after a telophase stage of mitosis result in increase in whole set of chromosome, whole set of chromosome in an organism. Okay, and this phenomena is known as polyploidy. Now, this polyploidy is often seen in plants and in case of any ploidy, in case of animals, polyploidy usually results in sterility. Okay, therefore polyploidy is rare in animal. Therefore polyploidy is rare in animals. Okay, so this is polyploidy. Increase in whole cell means suppose a diploid cell is there, it is converted into the tetraploid 4n, then it is a polyploidy. If it is a 3n, it is converted into 4n, then it is a polyploidy. Because after a nuclear division or karyokinesis, cytokinesis is not going to occur. Okay. So chromosome getting increased in the one cell itself. So because of this, it is a polyploidy. But now this polyploidy fall into two major categories. Means it is of two types. This polyploidy is of two types. One is autopolyploidy. And another one is allopolyploidy. So autopolyploidy and anopolyploidy, if we talk, what they are, we have to discuss this. Now in autopolyploidy or in case of allopolyploidy, there is only some difference. Don't confuse with that, like auto is associated with the autosomes and allo is associated with the allosomes or sex chromosomes. It is not like this. Here autopolyploidy means, here the term auto indicates that the polyploidy involves only homologous chromosome set. This ploidy involve only homologous chromosome set. Okay, so here what happens, the 3n is going to 6n, or particularly 2n is going to 4n. So just what is happening here, increase in the number of same genome. Here what is happening? Increase in the set of chromosome in one species itself. Then it is auto polyploidy. So if the 2n is there, diploid is there, and it becomes 4n, then it is autopolyploidy. Means the within a cell, a basic set is getting increased in the same species. Okay. So example, if we take of autopolyploid, then the examples are, okay, wheat, rice, or gram, we can say. Such type of autopolyploid can be increased. Means suppose the individual has a one into an A set and B set. A set having the 23 chromosome, B set having the 23 chromosome, and they are in a pair. Now, if this A set and B set both are getting duplicated, then it is going to be AABB. Okay. Then this condition is known as what? Auto, particularly we can say. Here, if we not take B, then it will be good to understand because we will be used to understand. Suppose they have AA2 set. This A represent one set, this A represent another set, this having the 23 chromosome, this having the 23 chromosome. Now this getting replicated. So it's going to be A. So whole two set become four set. Then it is an auto polyploid. If you want to draw a diagram of this, suppose this is two pairs are there. So, or we can say three pairs are there. So if three pairs are there, these three pairs having the six chromosome. Now when it get converted into the, so this is a two set. One whole set is increasing this side, one whole set is getting increased in this side. So this is going to be 4n. This three chromosome combined from one set. This another four will combine another set. Another four will another set. Another four another set. So four set are here, but here only two set are. So here increase in set of chromosome occurs, then it is autopolyploidy. But in allopolyploidy, it indicates what increase in the non-homologous set of chromosome. So this arises due to addition of, this arises due to crossing of, due to crossing of or fusion of gametes from two different species, then it is known as allopolyploid. Okay. So in allopolyploid, two different species are going to be involved, and they form a fusion and form a zygote, and that show polyploidy, then it is known as allopolyploidy. This allopolyploidy do not occur in nature. Actually, but however, some cytologists have produced allopolyploidy in certain plant by a selective breeding method. And the examples, if we talk about of allopolyploid, then there are number one, Raphenobrassica, and number two, that is Tritical. Now let's see how Raphenobrassica and Tritical are produced. First thing is that why it is known as Raphenobrassica. because it is made by a crossing of two different species, that is Raphanus sativus and Brassica oleracea. So let's take Raphanus sativus, that is radish, and Brassica oleracea. Okay. Now, when we see this Raphanus sativus, it is a diploid individual, and diploid individual have a 18 chromosome. So here 2n is equal to 18. Okay, and we represent its two set as a RR. Okay. Now here you will see that it is here 2n is equal to 18, and here it is a brassica, so we'll name a diploid one set B represent a one set, another B will represent another set. Now it produces when gametes, the gametes having a n is equal to 9, and that is having a one set, that is R. This gametes having a n is equal to 9, and that having a one set, that is B. Now when fusion of these two take place, the 2n is equal to 18, and this 2n is equal to 18 having a 9R and 9B, so we can write this as the RB. That is, R is the one set of this one Raphanus sativus, and B is representing a set of Brassica oleracea. Now in this, what autopolyploidy will occur, and in this when autopolyploidy will occur, it is known as allopolyploid. So it is 2n is equal to 18. So what will happen now? The 4n is going to be 36, and when 4n is going to be 36, this R set is going to be replicated, so it is going to be RRBB. Okay, so 18R will be there, 18B will be there, like this. Total 36 chromosome will be given to this, and this D is nothing but a Raphanus, that is allopolyploid, which is obtained like this. This diploid, we remember they are sterile hybrid, but this Raphanus can have a possibility of further twist. So this Raphanus have a large and vigorous plant body compared to the tetraploid. Same way, if we talk about the trickle, then this trickle is known as a new man-made cereal. Okay. Now in this trickle, the cross is made between Triticum aestivum and Cicale cereale. See, triticale can be hexaploid or octaploid. Okay. So if we take a first example of the hexaploid triticale, then we take a species that is Triticum durum, and it is crossing with the Cicale cereale. This having a 2n is equal to 28. This having a 2n is equal to 14. Now this Triticum durum, that is 2n is equal to 14, and is going to produce a gamete that is n is equal to 28. So it is going to be 14 in this gamete. Okay, and here its gamete going to have a 7 chromosome. Now why we are writing this as a 14 chromosome. So when F1 hybrid is produced, this F1 have a 2n is equal to 14 plus 7, that is 21, and this is the F1 hybrid, and this F1 hybrid is a sterile. Now further Colchicid treatment is given, then it particularly cause a polyploidy of this. So this 2n become what? 42. But if we see the polyploidy in this particular chromosome and 2n is equal to 42, then here this 42 is nothing but a what? We can say basic pair is 7 chromosome. So 7 6 are 42. So it is going to be a hexaploid critical. Total basic number are 7. So here total number of 42. So if one set has a 7 chromosome, then total 42. How many sets are there? 6 sets are there. So say hexaploid critical. But in another example, if we are taking a triticum estimum, and we are making a cross with the Cicalis relay, that is rye. This having a 2n is equal to 42. This having a 2n is equal to 14. The gametes will have a n is equal to 21. And here gamete will have a n is equal to 7. So it fertilizes and it will produce F1 hybrid which having a 2n is equal to 28. But this 28 F1 hybrid is going to be sterile. This is the F1. Now in this, when colchicine treatment given, this colchicine treatment will cause a doubling of chromosome. So it is going to be 2n is equal to 56. Yes, it is 2n is equal to 56, but with respect to, if we can say a basic number that is 7 in haploid, this is going having a 56. So 7 8 is 56. Okay, then it should be with respect to basic number of chromosome, it is going to be octaploid critical. Okay, this way the critical is produced. So here there is no need to how many chromosomes are there, and that is for the understanding. But you should remember the critical is produced by the hexaploid by the crossing triticum durum and Cicalis relay, and octaploid by the triticum estivum and Cicalis relay. Same way, Rapinobrassica is formed by the crossing between the two different species, that is the Raphana sativus and the Brassica oligarchy. And this way, this particular allopolyploid are produced. Okay, dear students. So I hope you understood this in detail, and that is all about the different type of mutation which are based on the cause. Okay. Now after this, there are some mutation on the basis, different bases. So now we are going to talk about another two different type of mutation based on the different base. So here next we are going to talk about a mutation, two type of mutation based on the type of cell, based on type of cell in which mutation occur. If we talk about this, then this is of two types. One is somatic mutation, and another one is the germinal mutation. In somatic mutation, the mutation occur in somatic cell. Mutation occur in somatic cell means that are the non-reproductive cell or body cells or cells other than the germinal cell or the non-reproductive cells. And this mutation, that is a somatic mutation, do not have any evolutionary importance. Okay. Whereas the germinal mutation occur in gametes or germ cells. Occur in gametes or germ cells. Then they are known as germinal mutation or gametic mutation. And this mutation are inheritable. Mean they pass from one generation to the another generation, and they are of great evolutionary significance. They are of great evolutionary significance. If the mutation are dominant, these are expressed in the next generation, and if recessive, the genotype expression remain suppressed. But this mutation will express in the next generation as well. That can be inherited. So this is the base where which type of cells are involved. Apart from this, mutation are of more to type based on different ways, and that is this mutation are of two types. In this, one is forward mutation, and one is backward mutation. If it is forward mutation, then there is a change from normal or wild type or dominant to new genotype. Not dominant, from a wild or normal original one to the new genotype. That new genotype can be either recessive or dominant. Then this is known as forward mutation, and this is the commonest type of mutation. Means common type of mutation. Whereas in backward mutation, an organism which has here an organism which has undergone forward mutation may again develop mutation which restore original wild type phenotype. Then this mutation or a reversal mutation is known as backward mutation or a reverse mutation. We can call it as a reverse mutation as well as. Why? Because it is re-establishing the original one. Okay. So this is the mutation, that is the forward and backward. Some more type of mutation, some terms are there, some more type of mutation are there, which you should remember. One is silent mutation. Then silent mutation, if we talk, they are the mutation in which base pair change in an allele's codon that produces no change in the amino acid sequence of a protein. Okay, so here what happens? Base pair change occur in alleles codon, but no change in amino acid, it is coding. Means genotype is getting changed, but phenotype is not getting affected or phenotype is not altered, then this type of mutation are known as silent mutation. Next mutation is known as missense mutation. In missense mutation, dear students, transition or translation of DNA base pair can give rise to the missense mutation. Why? Because in missense mutation, altered or changed amino acid codon gives a different amino acid, gives different amino acid in protein. So such protein products which are coded by the changed or altered gene produce partial activity and thus have a limited phenotypic change. Okay, then it is a missense mutation. They have a limited phenotypic change. And after this, another type of mutation are there which are known as nonsense mutation. In nonsense mutation, it occurs when an amino acid is changed to give a stop codon, that is UAA, UAG, or UGA, which do not code for any amino acid. So the polypeptide get terminated at this point and translation termination occur. Then it is known as nonsense mutation. What is happening? The normal gene is codon coding amino acid that is converted into the stop codon. So further, the translation will not continue and the new protein synthesis will stop. And some portion of protein is going to be getting produced. So nonsense mutation are usually extreme in this phenotypic and means totally it changes the particular whole phenotype. Okay, so these are the some of the particular different mutation which can be observed in the particular different genes, chromosome, or we can say number of chromosomes. Okay, so dear students, I hope you understood this topic in a very well manner. That is a mutation. It is some long topic, but easy to understand. Okay, so you can remember this, and you can, and there's some questions are asked from this particular point, so you will be able to understand that easily, and you can answer that easily. Okay, so till that, thank you.